All right, so today we're taking a deep dive into the world of immuno-oncology.
Yeah, immuno-oncology, where research is using the body's own defenses to, uh, you know, turn the tables on cancer.
Right, and we've got some recent papers to dig into, plus a really interesting trend analysis.
It's a fascinating field, really.
Definitely. So instead of just trying to wipe out cancer cells directly, we're kind of shifting gears. We're trying to figure out how to get the body to fight back on its own.
Exactly. And a lot of the research is focused on understanding how tumors, how they manage to evade the immune system in the first place. You see, cancer cells, they're tricky. They've developed these ways to evade. They essentially can hit the brakes on your immune system, making them invisible to your defenses.
So it's like cancer knows how to just vanish, even though our body should be able to see it.
Exactly. And we're uncovering more and more about how this disappearing act actually works. A good example of this is immune checkpoint molecules.
Ugh.
They're like the control panel for your immune response. But tumor cells, well, they can, hijack them, flip the off switch on your T cells, which are like, your body's elite cancer-fighting squad.
Okay, so hold on. These checkpoint molecules, they're like the brakes on the immune system. There's CTLA-4 and PD-1. They're supposed to keep things in check, make sure the immune system isn't like in overdrive all the time.
Exactly, yeah. Essential for balance.
But cancer messes that up.
Exactly. Slams on the brakes and stop T cells from doing their job.
So that's why cancer's been so tough to beat. It's been hiding in plain sight.
Yeah, essentially.
Okay, this is where it gets really interesting, at least for me. So scientists have developed these drugs called immune checkpoint inhibitors, ICIs, and they block those checkpoint molecules.
Right.
So the brakes are off, and the immune system can finally attack.
Yeah. It's been revolutionary in some cases.
Yeah.
Really effective for some patients.
This is what sparked all those headlines about miracle cures for cancer.
Yeah, it was a lot of excitement, especially with a class of drugs called antibody-drug conjugates, ADCs, hailed as potential magic bullets.
Right, yeah.
But while we have seen amazing success stories, with ICIs like Keytruda and Opdivo and FDA-approved ADCs like Kadcyla and Besponsa.
Not cure-all.
Not quite. It's not a cure-all. Not everyone responds to these therapies, and even when they do, the response isn't always, long-lasting. So a big focus of research now is figuring out how to make immunotherapy work for more people.
So how do we do that? What's the next step?
Well, think of it as Immunotherapy 2.0. One really exciting area is combination therapies, where we use different drugs together to boost the immune response. It's like we're trying to fine-tune the strategy, right? So instead of just one approach, we're saying, okay, what happens if we try this, this, and this? What if we combine different approaches?
Right, like trying to find the right combination to really pack a punch.
Exactly. And some clinical trials have combined checkpoint inhibitors like ipilimumab, which targets that CTLA-4 we talked about, with nivolumab, which targets PD-1.
So kind of like a double whammy to really unleash the immune system.
Yeah, you could think of it that way. And the idea is that by targeting multiple checkpoints, we can really amplify that immune response, make it even stronger.
And have these trials shown promise?
They have. But, and this is important, it's not a one-size-fits-all solution. The CheckMate trials, which looked at this combination, have shown some really remarkable success in certain cancers, like melanoma.
Yeah.
But the results have been, well, a little less consistent in others, like colorectal or urothelial carcinoma.
So some cancers might be more susceptible to this kind of multi-pronged attack than others.
It seems that way, yeah.
But I imagine combination therapies come with their own, their own set of challenges, right? It's not always easy to just throw a bunch of drugs together.
You're absolutely right. There's always a balance. On the one hand, combination therapies offer this potential for real enhanced efficacy, but they also carry the risk of increased side effects.
So it's about finding that sweet spot where it's effective but also safe.
Exactly. Finding that balance is crucial.
Okay, so besides combination therapies, what other strategies are researchers exploring in this Immunotherapy 2.0 toolkit?
Well, we talked about releasing the brakes with checkpoint inhibitors, but there's also this idea of hitting the gas pedal at the same time. That's where costimulatory agonists come in.
Okay, so instead of just taking your foot off the brake, you're also stepping on the gas.
Exactly. A perfect analogy. And these costimulatory agonists, they activate certain molecules that stimulate the immune system, give those T cells an extra boost. One promising target for these costimulatory agonists is a molecule called 4-1BB.
4-1BB?
Yeah, so activating 4-1BB can really enhance T-cell proliferation and survival, leading to a more potent and hopefully long-lasting immune response.
That sounds really promising. But I'm guessing there are challenges with this approach, too.
You're right to be cautious. While promising, 4-1BB agonists have been associated with liver toxicity in some cases. So researchers are working on ways to kind of refine this approach, improve its safety and make sure it's as effective as possible.
That makes sense. What are some of the strategies they're looking at?
One really interesting avenue is the development of targeted delivery systems.
Okay, yeah, I've heard a bit about that.
So imagine a system that can deliver the drug directly to the tumor, minimizing any off-target effects.
So less damage to healthy tissues.
Exactly. It's like delivering a guided missile precisely to its target.
Wow, that level of precision is amazing.
It really is. And there's another layer to this I think you'll find fascinating. You see, for a long time, the focus of immunotherapy was really on T cells.
Right.
But we're realizing now that other immune cells play a really vital role in fighting cancer as well.
Oh, really? So it's not just a one-man army?
Exactly. It takes a coordinated immune response. Natural killer cells, or NK cells, they're another type of immune cell that can directly kill tumor cells. They act like your body's first line of defense.
That's incredible. So we've got these different players on the field, all with their own unique skills to tackle cancer.
Exactly. And there's one more I want to mention, though. They're a bit more complex. They're called tumor-associated macrophages, or TAMs.
TAMs, okay.
And these TAMs, they're immune cells that are actually found within the tumor microenvironment. And they can be a bit of a double-edged sword.
How so?
Well, they can either help or hinder tumor growth depending on the signals they receive from that tumor microenvironment. Think of it like a tug-of-war.
Okay, I see.
In some cases, they might suppress the immune response, which unfortunately helps the tumor survive. But in other cases, they can actually be activated to promote an anti-tumor response.
Wow, so these TAMs could be powerful allies if we can figure out how to, shift their balance in our favor.
Precisely. And that's where a lot of research is focused right now. If we can crack the code of how to manipulate TAMs, it could be a real game changer for cancer treatment.
It's like we're learning to orchestrate this complex symphony of immune cells. It really is like we're learning to conduct this whole orchestra of immune cells inside the body, each one playing a specific part to get rid of the cancer. And you mentioned targeted delivery earlier and how important that is?
Yeah, targeted delivery is really key here. Scientists are developing these things called nanoparticles.
Okay, yeah, I've heard of those. They're like, what? Incredibly tiny.
Yeah, incredibly tiny particles, and they act like Trojan horses. They deliver drugs directly to the tumor, so there's less collateral damage, less damage to the healthy tissue.
Like a guided missile.
Exactly.
But I bet it's hard to create something that small and that complex.
Oh, absolutely. It's really cutting-edge science.
Yeah.
And there are different types of nanoparticles, each with its own strengths and weaknesses.
Oh, really? So it's not just one type fits all?
No, not at all. Some are made from lipids, kind of like what's used in the mRNA vaccines.
Right.
Others are metallic, made from gold or even silver.
Wow.
And then there are these really interesting ones called mesoporous silica nanoparticles.
What was that last one?
Mesoporous silica nanoparticles.
Okay.
And they have this porous structure, like a tiny sponge, that lets them carry a lot of the drug.
So how do these nanoparticles know to go to the tumor? How do they actually find it?
That's a great question. So some nanoparticles use something called passive targeting. Tumors often have leaky blood vessels, and these nanoparticles are so small that they can slip through the gaps and just build up in the tumor.
Ah, so they're using the tumor's own weaknesses against it.
Exactly. But it gets even cooler. Researchers are also developing nanoparticles that use active targeting, and these are decorated with, imagine them with these tiny homing beacons. They're molecules that bind specifically to the receptors on tumor cells.
Like a GPS system to find the target? That's amazing.
It really is remarkable. And it's not just about building the tools, right? It's also knowing how to use them. It goes back to what we've said before about personalized medicine.
Right. The right therapy for the right person at the right time.
Exactly. We need to know which patients will benefit from these therapies. And that's where biomarkers come in.
Biomarkers?
Yeah, think of them like molecular fingerprints.
Okay.
They can reveal things about the tumor, the genetic makeup, the proteins it's expressing, all of that.
So doctors could use that information to see who would respond to which immunotherapy.
Precisely. And they're finding more and more of these biomarkers, like there are proteins called ENO1, Nectin-4, and TROB2 that look really promising.
So we're getting closer and closer to really figuring this all out.
We are. And while those established targets like PD-1 and PD-L1 are still important, there's more and more focus on these newer targets like TIGIT, B7H3, and LAG3. They're all immune checkpoint molecules.
And they could be targets for new therapies.
Exactly. And it's not just about these checkpoint molecules. There's a lot of excitement around CORE-NK cells.
Core RNK cell.
Yeah. They combine CORA T cell therapy, which I'm sure you've heard of, with natural killer cells.
So you're taking the best of both worlds.
Yeah, that's the idea. And then there's RNA-based vaccines.
Right, like COVID vaccines.
Exactly. They can be huge in training the immune system to recognize and attack specific tumor antigens.
So the immune system basically learns to hunt down the cancer cells more effectively.
Yep, that's the goal.
It's amazing how far we've come. We've gone from just understanding how the immune system works to actually manipulating it to fight cancer.
It's truly incredible. But we still have work to do. A lot of patients still don't respond to immunotherapy, and that's a big challenge.
Yeah, I imagine so.
One of the big questions is whether we can crack the code, so to speak, of these cold tumors.
Cold tumors.
Someones that are resistant to immunotherapy.
Yeah.
If we can turn them hot, make them susceptible to the immune system, that would be huge.
It seems like there's so much potential in this field. It's really inspiring.
It really is.
Well, that's about all the time we have for today. It's been a fascinating deep dive into the world of immuno-oncology. Thanks for joining us.